Hello, sir.
Hello, good evening, madam.
Good evening, sir.
Yeah, please proceed, madam. Immunology, part two.
Okay, sir.
Yes, madam.
Hello, everyone. Welcome to Tawhid Foundation for Allied Healthcare Sciences, Bangalore. Today's class is about Immunology Part 2 for BSc MLT third year students. Today, we'll discuss about antigen and antigen recognition molecules. So, here now we are going to explore one of the most fundamental concept in immunology, that is antigen. Antigen are the substances that trigger our immune system to respond and protect the body against infections, foreign molecules, or abnormal cells. Understanding this antigen is crucial because they form the basis of how our immune system recognizes threats, produces antibodies, and develops immunity. So in today's class, we will cover the definition of antigen, their types, how they are recognized by B cells and T cells, the structure and function of antibodies, and the role of major histocompatibility complex molecules. So let's begin with the definition and basic concepts of antigen. An antigen is defined as any substance that has the ability to provoke the immune system of an animal or human being to respond by producing an immune reaction or responses. What makes an antigen unique is that this reaction is directed specifically against the antigen itself and not against unrelated substances. So the immune response may sometimes involve only the humoral arm of the immune system, which includes antibodies, B lymphocytes, or only the cellular arm, which include T lymphocytes. However, in most cases, both arms of the immune system are activated together. It is also important to emphasize that not all immune cells take part in the response. Only those B cells and T cells clones whose surface receptors specifically match the antigen will be stimulated. For B cell, these receptors are membrane-bound immunoglobulins, and for T cells, the receptors are T cell receptor TCR proteins. So this means the immune system is highly selective. It reacts only when a receptor and antigen fit together like a lock and key system. Recognition of antigen. The way B cells and T cells recognize antigens is fundamentally different. B cell will recognize soluble antigens directly. The surface receptors are membrane-bound immunoglobulins, also called B cell receptors or BCR. When an antigen in its soluble form binds to the BCR, it activates the B cell. This binding does not require processing or presentation by other cells. T cells, however, cannot recognize free antigens. Instead, they recognize antigens in the form of short peptides, fragments that are bound to major histocompatibility complex, that is MHC, molecules on the surface of antigen presenting cell, that is APCs. Such macrophages, dendritic cells, and B cells. In other words, the T cell epitope is actually a combination of the peptide antigen plus the MHC molecule. Therefore, in the strictest immunological sense, an antigen can be defined as a substance that is recognized either by the immunoglobulin receptor of a B cell or by a T cell receptor, that is TCR, which is associated with an MHC molecule. Now, what is epitopes? Antigen receptors do not recognize the whole antigen molecule. Instead, they recognize discrete region on the antigen known as epitopes or antigenic determinants. An epitope is the smallest region or smallest structural unit of an antigen that can be recognized by an antibody or a T cell receptor. Different lymphocytes, each with a unique receptor, recognize different epitopes on the same antigen. This is why a single antigen can stimulate a broad and diverse immune response. For B cell, the B cell receptor can recognize epitopes in many forms. They may be present on free soluble antigens or on surface bound molecules or even as degraded fragments of larger molecules. So, in contrast, the T cell receptor is much more restricted. It can only recognize epitopes that consist of peptide fragments bound to MHC molecules on the surface of antigen presenting cells. So, based on the type of immune responses they generate, antigens and their epitopes can be divided into three broad functional categories, that is, immunogen, haptens, and tolerogens. This classification will highlight that not only all antigen behave the same way in the immune system. So these are the three categories, functional categories of antigen. First is immunogens, that is complete antigen. It induces immune response. Second is haptens. It is partial antigens, that is antigenic but not immunogenic alone. Third is tolerogens, that is usually self-induced immune unresponsiveness. So we'll go one by one what exactly it is. We'll start with immunogens, that is nothing but complete antigens. Immunogens are antigens that are capable of inducing a full immune response. They stimulate the production of antibodies or activate sensitized T lymphocytes, which then react specifically with the immunogen that triggered them. A key principle here is all immunogens are antigens, but not all antigens are immunogens. This is because immunogenicity refers to the ability to provoke an immune response, while antigenicity refers only to the ability to bind to immune receptors. For example, large proteins and complex polysaccharides are often immunogenic because they are large and chemically complex, allowing the immune system to detect them as foreign. In contrast, very small molecules may be antigenic. They can bind to antibodies, but by themselves, they do not stimulate an immune response. They are known as haptens, which we will discuss next. Now, haptens. Haptens are partial antigens. Haptens are small molecules of low molecular weight that are antigenic but not immunogenic on their own. This means that they can react with antibodies but cannot independently stimulate the production of antibodies. For a hapten to become immunogenic, it must be attached to a larger carrier protein. Once bound, the combined molecule behave as a complete antigen that can trigger an immune response. So a classic example is a penicillin allergy. Penicillin by itself is too small to stimulate an immune response. However, in some individuals, it binds to serum proteins and forms a hapten carrier complex. So this new complex is immunogenic and triggers the production of antibodies. In these individuals, exposure to penicillin again causes the antibodies to react with penicillin, leading to hypersensitivity or allergic reactions. Therefore, haptens illustrate the important distinction between antigenicity, that is the ability to bind, and immunogenicity, the ability to stimulate a response. So as you can see in this diagram, haptens are very small molecules that by themselves cannot stimulate an immune response. They become immunogenic only when attached to a larger carrier protein. Example, as I mentioned earlier, penicillin act as a haptens binding to proteins and causing allergy. Here, stress concept, hapten alone is equals to non-immunogenic. Hapten plus carrier is equals to complete antigen. So, hapten alone, non-immunogenic, and then hapten molecules plus carrier is equals to complete antigenic. Next is tolerance. So tolerogens are antigens that under normal physical conditions do not provoke an immune response but instead induce immune unresponsiveness. So most tolerogens are self antigens during the development of the immune system. Particularly in early life, the body learns to tolerate its own molecules. This process is called immunotolerance and ensures that the immune system does not attack self tissues. For example, proteins in our own cells, such as enzymes or structural proteins, do not normally provoke an immune response. However, if tolerance fails, the immune system may mistakenly identify these self molecules as foreign and mount a response against them, leading to autoimmune diseases. Thus, tolerogens are essential in maintaining the balance between defense against foreign invaders and protection of self tissues. Next is classification of antigens based on origin and microbial antigens. Antigens can also be classified according to their origin. Microbial antigens are among the most important. This can be further divided into different types based on their location or function within the microbe. So there is different types: febrile, somatic, flagellar, and capsular. So we'll see one by one. Febrile antigens are located on the fimbriae of bacteria and play a role in bacterial attachment to host cells. Somatic antigens are structural components of the bacterial cell wall. Flagellar antigens are associated with bacterial flagella and are important for motility. Capsular antigens are found in the polysaccharide capsules surrounding some bacteria, which help them evade immune defenses like phagocytosis. So each of these antigens has different types, distinct immunological importance, and the immune system can generate specific responses to target them. So classification based on immune response. Antigens can also be classified based on this type of immune response they produce. First is T independent antigens and T dependent antigens. T independent antigens are antigens that can stimulate B cells to produce antibodies without the help of T cells. They are usually large molecules with repeating epitopes, such as polysaccharides or lipopolysaccharides found in bacterial cell walls. These antigens primarily induce the production of IgM, immunoglobulin M, antibodies and do not generate strong memory responses. As a result, the immunity they provide is often short-lived. So T-independent antigen, it will stimulate B cell without T cell help. It has large molecules with repeating epitopes and it produces mainly IgM and short memory. Second is T-dependent antigens. On the other hand, it required the assistance of T cells to stimulate B cells. This include proteins, red blood cell antigens, and hapten carrier complexes. T-dependent antigens induce a more robust response, leading to the production of multiple classes of antibodies such as IgM, IgG, IgA, and IgE. They also generate long-term immunological memory, which is the basis for effective vaccination. This distinction between T-independent and T-dependent antigen is very important in immunology because it explains why some antigens generate strong and long-lasting immunity while others do not. Factors determining immunogenicity. Not all antigens are equally effective in stimulating the immune system. The degree to which a substance can trigger an immune response is called immunogenicity, and it is determined by several important factors, that is molecular weight, chemical complexity, and foreignness. Molecular weight. In general, larger molecules are more immunogenic than smaller ones. Proteins with high molecular weight, such as hemocyanins, which have a molecular weight of 6.75 million, are highly immunogenic. By contrast, molecules smaller than 10,000 daltons are weakly immunogenic. These smaller molecules can only stimulate an immune response if they are linked to larger carrier proteins, which is the principle behind haptens. Second is chemical complexity. Proteins and polysaccharides are strong immunogens because they are chemically complex, containing multiple different building blocks arranged in diverse structures. In contrast, lipids and nucleic acids are poor immunogens unless they are presented as part of complex structures. Even among proteins, not all are antigenic. Gelatin, for example, is not immunogenic because it lacks aromatic amino acid like tyrosine, which are essential for antigenic recognition. Third is foreignness. One of the fundamental job of the immune system is to distinguish between the self and non-self. The more different an antigen is from host molecules, the stronger the immune response will be. If an antigen closely resembles self tissues, the immune system may ignore it or react only weakly. Next, other factors is epitopes. So presence of epitopes, that is antigenic determinants. The number and accessibility of epitopes on an antigen also affect immunogenicity. More epitope means more opportunities for lymphocyte to recognize the antigen, increasing the immune response. Next is genetic constitution of the host. Not all individuals respond to antigen in the same way. Two members of the same species may react differently because of differences in their immune response genes. This is why some people are more resistant or more susceptible to certain infections than others. Next is susceptibility to tissue enzymes. Antigens must be processed, broken down, and presented to the immune system. Substances that are easily degradable or degraded by tissue enzymes are more likely to be immunogenic. In contrast, synthetic molecules like polystyrene, latex, or certain polypeptides that are resistant to enzymatic breakdown are usually non-immunogenic. Next is dosage, route, and timing of administration. The way an antigen enters the body matters. For example, oral, intravenous, or intramuscular routes of administration can produce different immune responses. The dose also influences the outcome. Too small a dose may fail to stimulate a response, while too large a dose may induce tolerance. Timing or repeated exposure also play a role in shaping the response. Next is use of adjuvants. Immunogenicity can be artificially enhanced by mixing the antigen with an adjuvant. Adjuvants work by maintaining the antigen in the body for a longer period, ensuring slow and continuous stimulation of immune cells. They also enhance the recruitment of immune cells to the site of injection. This is why vaccines often contain adjuvants like aluminum salts. So, in summary, immunogenicity is not determined by a single factor but by a combination of size, complexity, foreignness, host genetics, degradability, dose, route, and the use of adjuvants. Next is antigen receptors. Antigen recognition is carried out by specific receptors, and these differs between the adaptive and the innate immune system. In an adaptive immunity, the key receptors are B cell receptors, that is BCRs, and T cell receptors, that is TCR. These are generated somatically, meaning each individual B cell or T cells has a unique receptor specific for a particular epitope. BCRs are essentially membrane-bound immunoglobulins that can bind to soluble or native antigens directly. TCRs, in contrast, recognize antigens only when they are presented as peptide MHC complexes on the surface of antigen-presenting cells. Next is antigen receptors and innate immunity. In innate immunity, antigen recognition does not rely on somatically generated receptors, but on preformed receptors that recognize broad, conserved microbial structures. These are called pattern recognition receptors, that is PRRS. PRRs are designed to detect pathogen associated molecular patterns, that is PAMPs, which are characteristic molecules found in microbes but absent in host tissues. Example of soluble PRRs include mannose binding lectin, that is MBL, C-reactive protein, that is CRP, and lipopolysaccharide binding protein, that is LPP, and complement proteins. These circulate in the blood and tissue fluids and help opsonize pathogens. Next is cell surface PRRs. On cell surface, PRRs include scavenger receptor, that is SRs, which bind bacteria and apoptic cells, and toll-like receptor, that is TLRs, which are perhaps the most important PRRs. TLRs trigger the production of cytokines, promoting inflammation and directing immune cells to the site of infection. Other innate receptors like killer activation receptors, that is KAR, and killer inhibition receptor, that is KIR, on natural killer cells, as well as complement receptors and FC receptors on phagocytes. These receptors ensure that the innate immune system can mount a rapid, though non-specific response, while the adaptive immune system gears up its highly specific defense. Next is antigenic specificity. The specificity of antigen recognition is a fascinating aspect of immunology. The basis of antigen specificity is stereochemical, meaning that the shape and spatial arrangement of chemical groups on the antigen determine whether or not the immune system can recognize it. Even a single chemical grouping or the position of a functional group, whether ortho, meta, or para, can alter the specificity of an antigen. However, antigenic specificity is not absolute. Sometimes cross-reaction occurs where antibodies or T cells generated against one antigen also recognize another structurally similar antigen. This has important clinical consequences. For example, antibodies produced against streptococcal bacteria can cause cross-react with human heart tissue, leading to rheumatic fever. So this is the diagram of this antigenic specificity. Specificity refers to the exact fit between antibody and antigen determined by unique antigenic determinants, that is epitopes. Even slight structural changes can prevent recognition. Example is blood group antigens, and why transfusion must be exact. Next is types of specificity. The types of antigenic specificity can be grouped as follows. Specific specificity: all individuals of a species share certain antigens. For example, tissues from one species can be distinguished from another based on species-specific antigens. This is useful in evolutionary history studies and forensic science. Next is isospecificity. Isoantigens are present in some individuals but not all within a species. The best example is the ABO blood group systems. These isoantigens are genetically determined and are crucial in blood transfusion and paternity testing. Next is autospecificity. Self-antigens are normally tolerated, but under certain conditions, they may provoke an immune response. For example, lens proteins are normally hidden from the immune system. But if the lens capsule is damaged, these proteins can be exposed and provoke an autoimmune response. Similarly, sperm cells which can develop after immune tolerance is established can act as antigens if they enter circulation due to injury. Next is organ specificity. Some antigens are shared across the same organs of different species. For instance, brain or kidney antigens may cross-react across species. A clinical example is neuroparalytic complications that occur after the old ship brain-derived rabies vaccines due to cross-reaction of brain antigens. Next is heterogenic, that is heterophile specificity. Some antigens are shared across very different species, including animals, plants, and bacteria. These are called heterophile antigens. A well-known example is the Forceman antigen, a lipid carbohydrate complex found widely across species. Clinically, heterophile antigens are used in diagnostic tests such as the Weil-Flix test for typhus fever, the Paul Bunnell test for infectious mononucleosis, the cold agglutinin test for mycoplasma pneumonia infection, and the cardiolipin antigen test for syphilis. These are the example of this heterophile antigen. As I mentioned earlier, Forceman antigen, it is widely distributed. Proteus oxstrains, diagnosis of typhus. Through Weil-Felix test, Ship's RBCs, diagnosis of infectious mononucleosis, that is Paul Bunnell's test. Cold agglutinin test, that is mycoplasma pneumonia, and cardiolipin antigen, syphilis, STDs. So, in addition to the conventional tissue specific antigen, these are certain antigens called heterophile antigens. These antigens are interesting because they are structurally similar across different species, classes, or even kingdoms, which allow for cross reactivity in immune tests. A classic example is the Forceman antigen, a lipid carbohydrate complex that is widely distributed in humans, animals, birds, plants, and even some bacteria. Its widespread presence makes it a useful model for studying cross-reactive immune responses. Heterophile antigens have important diagnostic applications in clinical serology. For instance, Proteus OX strains such as OX19, OX2, and OXK are used in the Weil-Felix test to diagnose typhus fever. This test relies on the cross-reactivity between Proteus antigen and antibodies produced during infection with Rickettsia species. Similarly, Ship RBCs, red blood cells, are used in the Paul Bunnell test to detect infectious mononucleosis caused by the Epstein-Barr virus. In this test, antibodies in the patient's serum agglutinate the ship's RBCs due to cross-reactivity with heterophile antigens. Another example is cold agglutinin test, which is used to identify infection caused by mycoplasma pneumonia. Infected individuals produce antibodies that bind to RBCs at low temperatures, causing them to agglutinate. Lastly, the cardiolipin antigen is used in the serological test for syphilis, that is STS. This test detects antibodies against cardiolipin, a lipid component that cross-reacts with Trypanosoma pallidum infection. So overall, heterophile antigen and these diagnostic antigens illustrate how antigenic cross-reactivity can be harnessed to detect infections that are otherwise difficult to identify directly. These tests are simple, rapid, and clinically valuable, making them important tools in medical microbiology. Thus, antigenic specificity highlights both the precision and the occasional cross-reactivity of the immune system. Next is antigen recognition molecules. The immune system requires precise recognition mechanism to distinguish between self and non-self. This recognition is made possible by specialized molecules. So, Antibodies, immunoglobulin IG. Antibodies exist in two forms. First, as membrane-bound immunoglobulins on B cell surfaces, where they act as B cell receptors, that is BCRs. These receptors consist of immunoglobulin molecule closely associated with signaling proteins like Ig alpha and Ig beta. Second, antibodies are secreted in soluble forms by plasma cells. These circulating antibodies in serums and tissues are structurally identical to BCRs but lack the transmembrane and cytoplasmic portions. So their role is to bind antigens with high specificity, neutralize pathogens, and recruit other immune mechanisms. So in antibodies, there are two types. Okay. And second is T cell receptor, that is TCRs. It is found only on T cells, that is TCRs are membrane bound receptors that recognize peptides presented by MHC molecules. Unlike antibodies, TCRs cannot recognize soluble antigen. They are absolutely dependent on the antigen being processed into small peptides and displayed by an MHC molecules. Third is major histocompatibility complex, that is MHC. MHC molecules are essential for antigen presentation. Class 1 MHC molecules present peptides from proteins synthesized inside the cell, such as viral proteins. Class 2 MHC molecules present peptides from proteins taken up from outside the cell, such as bacterial proteins. This division ensures that the immune system can respond to both intracellular and extracellular pathogens. So all these molecules, antibodies, BCRs, TCRs, and MHC proteins share a common structural feature known as immunoglobulin fold, that is Ig fold. This is why they belong to the Ig supreme family. So as you can see in this diagram, Ig supreme family, which also includes other proteins such as cell adhesion molecules, B2 microglobulin, and platelet derived growth factor receptors. So in this figure, some members of the immunoglobulin supreme supergene family, a group of structurally related, usually membrane bound glycoproteins. This diagram shows Ig supreme family members. Example is Ig TCR, that is T cell receptor, MHC, that is major histocompatibility complex molecules. They all share the immunoglobulin fold structure. Evolutionary conservation show that their role in antigen recognition and immune signaling, and not all antibodies, some are cell surface receptors and presentation molecules. Next is antibodies. Antibodies, also known as immunoglobulins, are the effector molecule of humoral immunity. They are proteins produced by B cells and plasma cells in response to antigens, and they function to recognize and neutralize foreign substances such as bacteria, viruses, and toxins. Antibodies exist in two distinct forms. The first form is B cell receptors, BCR, which is membrane bound on the surface of B lymphocyte. The BCR is essentially an immunoglobulin molecule attached to the B cell membrane, and it is closely associated with the signaling heterodimer known as Ig alpha and Ig beta. This heterodimer is crucial because it transmits activation signals into the B cell when the receptor binds its specific antigen. This signaling triggers B cell activation, proliferation, and differentiation into antibody producing plasma cells or memory B cells. Second form is secreted form. The second form in the secreted antibody, which is released from plasma cells into the serum and other body fluids, while soluble antibodies and BCRs are structurally identical in terms of their antigen binding regions. The key difference is that secreted antibodies lack the transmembrane and cytoplasmic region. This allows them to circulate freely in blood, lymph, and extracellular fluids, where they can neutralize pathogen, opsonize microbes for phagocytosis, active complement, and perform other immune functions. So in essence, the PCR serves as the sentinel on B cell, detecting antigen and initiating immune responses, whereas the secreted antibody serve as the effector molecule performing immune functions at a distance from the B cell. Both of these forms together ensures that the humoral immune system can recognize antigen, initiate a response, and eliminate threats efficiently. Next is antibody history. The history of antibody discovery begins with observation in the 18th century, when it was noted that the injection of an antigen into an animal resulted in the appearance of specific substances in serum that could react with that antigen. These substances were called antibodies. Depending on their functions, they were referred to as agglutinins, precipitants, or complement fixing antibodies. Later, fractionation of immune sera revealed that antibodies belong to a specific group of proteins called globulin. Using electrophoresis, serum proteins were separated into albumin, alpha, beta, and gamma globulin. Antibody activity was found in the gamma globulin fraction. This is why antibodies are sometimes called gamma globulin. In 1964, a common terminology was adopted intentionally, the term immunoglobulin Ig. Immunoglobulins are defined as animal proteins with known antibody activity as well as related proteins found in certain pathological conditions like myeloma or macroglobulinemia. These comparative features of immunoglobulin isotypes. In this table, it compares IgG, IgA, IgM, IgD, and IgE. It highlights molecular weight, serum concentration, and biological distribution. IgG, most abundant, it crosses placenta. IgA, it protects mucosal surfaces. IgM, it is first responder, that is pentameric. IgD, it is B cell receptor. IgE, allergy and parasite defense. You can go through this table for comparative features of immunoglobulin isotypes. Next is principal biological activities. It summarizes functions of immunoglobulin classes: IgG, opsonization, complement activation, neonatal immunity; IgA, mucosal defense, prevents pathogen adhesion; IgM, complement activation, agglutination; IgE, allergy, parasite immunity; IgD, role in B cell activation. You can refer this table for principal biological activities. Next is IgG structure, schematic representation of an IgG molecule, indicating the location of the constant and the variable regions on the light and heavy chains. So here the diagram shows IgG-Y. As you can see, it is a Y-shaped molecule. Variable region, tips of Y, as you can see, binds antigens. Constant region, FC, mediated effector functions, heavy chains, that is 50 kDa each, light chains 25 kDa each. Important dual role, antigen binding plus immune system activation. This diagram is important for exam purpose. Next is antibody definition. So the distinction between antibody and immunoglobulin is subtle. All antibodies are immunoglobulins, but not all immunoglobulins are antibodies. For example, abnormal proteins like myeloma proteins are immunoglobulins, but not true functional antibodies, as you can see here. Next is structure of Ig molecule. All immunoglobulin are comprised of two light chains and two heavy chains linked together by disulfide bonds to form a Y-shaped structure. Light chains about 25 kDa are either of the kappa or lambda type. Heavy chains about 50 kDa are unique to each classes of immunoglobulin and determine the class gamma Y for IgG mu, for IgM alpha, for IgA delta, and epsilon, that is for IgE. So gamma, mu, alpha, delta, and epsilon for different immunoglobulin. The antigen binding site is formed by the variable regions of both the light and heavy chains located at the amino terminal and the carboxy terminal portion of the heavy chain forms the Fc region, which is responsible for biological functions such as complement activation and binding to Fc receptors on immune cells. Next is fragment of Ig by enzyme digestion. So proteolytic cleavage of IgG provide further structural insight. Papain digestion yield two Fab fragments which contain antigen binding sites and one Fc fragment. Pepsin digestion produces a bivalent FAB2 fragment that can bind antigen but lacks Fc function. Fc mediate complement activation, macrophage binding, and cell receptor binding. Next is hypervariable regions. Both light and heavy chains are divided into variable regions and constant regions. The variable regions from the highly diverse antigen binding site, while the constant region mediate effector functions. Within the variable regions, three hypervariable regions exist known as complementary determining region, that is CDRs, which form the precise antigen binding interface. Classes of immunoglobulins. Immunoglobulin G, IgG, is the most abundant antibody in serum, making up 70 to 75% for 70 to 75% of total immunoglobulin. It exists as a monomer and has a molecular weight of around 150 kDa. IgG is a major component of the secondary immune response, making it dominates after repeated exposure to an antigen. So unique features of IgG include it has the longest half-life of about 23 days, making it crucial for long-term immunity. It is the only antibody class that can cross the placenta, providing passive immunity to the fetus and newborn. It participates in key immune reactions, complement activation, especially Ig1 and Ig3, opsonization, neutralization of toxins and viruses, and antibody-dependent cellular toxicity, that is ADT. So there are four subclasses of IgG in humans, that is Ig1, Ig2, Ig3, and Ig4. These differ slightly in their heavy chain structure and functional properties. Ig1 and Ig3 are the strongest in complement activation and Fc receptor binding. This is the schematic representation of proteolytic digestion of immunoglobulin G. IgG pepsin treatment produces a dimeric FAB2 fragment. Papain treatment produces monovalent Fab fragments and an Fc fragment. The FAB2 and the Fab fragments bind antigen but lack a functional Fc region. So as you can see in this picture, papain clave produces the two Fab fragment, that is monovalent plus one Fc. Pepsin cleavage produces one FAB2 fragment, that is bivalent Fc degraded, and Fab binds antigen Fc needed for biological function. Application: Fab fragment used in diagnostic theory. So for human IgG subclasses, IgG1, IgG2, and IgG3 and 4 shown with different hinge regions. These IgG1, IgG2, IgG3, and IgG4 for human IgG subclasses with differences in hinge regions and disulfide bonds. IgG3 has longest hinge and 11 disulfide bonds, as you can count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So 11 disulfide bond, more flexible but less stable. Subclasses differ in placental transfer and complement activation. Clinical importance, it is selective subclass deficiency can cause immunodeficiency. This next classes of immunoglobulin IgA. IgA is the second most abundant immunoglobulin in serum, but is most important at mucosal surfaces. It makes up about 10 to 13% of total serum immunoglobulin. In serum, IgA is found mostly as a monomer. At mucosal surface, that is respiratory tract, gastrointestinal tract, urogenital tract, IgA exists as a dimer with a joining J chain and a secretory component forming secretory IgA. So SIgA, that is secretory IgA, is the main antibody in secretions such as saliva, tears, breast milk, and mucosal secretions. It plays a critical role in mucosal immunity, preventing pathogens from adhering to epithelial cells and neutralizing toxins. IgA does not cross the placenta, but breast milk provides IgA to newborns, protecting them against gastrointestinal infections. Functionally, IgA promotes phagocytosis, prevents colonization by pathogens, and can activate the alternative complement pathway. So as IgA does not cross the placenta, but breast milk provides IgA to newborn, which protect them against gastrointestinal infection. As you can see, for newborn, it is not allowed to give outside milk. That is why breast milk is provided to avoid children from gastrointestinal infection. This is the structure of IgA. It exists as a monomer in serum and dimer as secretory IgA in mucosa. Dimer joined by J chain, as you can see, plus secretory component. It protects against enzymatic degradation, major defense in respiratory and gastrointestinal tract. Next is IgM. IgM is the first antibody produced during the primary immune response. It makes around 5 to 8% of serum immunoglobulins and has a relatively short half-life of 5 days. Structurally, IgM is a pentamer consisting of 5 immunoglobulin units linked by disulfide bonds and a J chain. This gives IgM a very high valency with 10 potential antigen binding sites, though steric hindrance allows fewer to actually bind simultaneously. Because of its large size, IgM is confined mostly to the bloodstream. It is extremely efficient at activating complement and agglutinating pathogens. Isohemagglutinins, anti-A and anti-B blood group antibodies are naturally occurring IgM antibodies. Importantly, detection of IgM in a fetus or newborn suggests intrauterine infection since maternal IgM does not cross the placenta. Structure of human IgM pentamer. It is the largest antibody pentameric with J chain, highly efficient at agglutination and complement activation. It is the first antibody to appear in primary immune response, and clinical significance of IgM levels indicate recent infections. Next is IgD. It is present in trace amounts in serums around 0.03 mg per ml. Its half-life is only about 3 days. Although it is not abundant, IgD has an important role as a B cell receptor. On immature B cell, IgD along with IgM serve as surface receptors for antigen recognition, helping to initiate B cell activation. So structurally, IgD resembles IgG but lacks some effectors functions. It is not typically secreted in large amounts and does not fix complement. This is the structure of IgD. It is very low serum concentration. It is found mainly on surface of immature B cells as BCR. It helps initiate B cell activation. Its exact physiological role still under study. Next is IgE. IgE immunoglobulin is found in very low concentration in serum, but it has unique biological functions. It binds with high affinity to Fc receptors on mast cells and basophils. When IgE encounters its specific antigens such as pollen, food protein, or helminthin antigen, it cross-links receptors on these cells, triggering the release of histamine and other mediators. So these lead to immediate hypersensitivity reactions such as anaphylaxis, asthma, and allergic rhinitis. IgE also plays a role in defense against helminthic infections. This is the structure of IgE immunoglobulin. It is very low serum levels but highly potent. It binds strongly to Fc receptors, as you can see in this diagram, on mast cells and basophils. It mediates allergic reactions, histamine release after allergen binding. It is also important in defense against helminthic parasites. Next is abnormal immunoglobulins. In certain pathological conditions, like multiple myeloma, plasma cells produce abnormal monoclonal immunoglobulins. These are structurally similar to normal antibodies but are produced in excess by a single clone of cells. So, Bence-Jones proteins, for example, are free light chain excreted in the urine of myeloma patients. Depending on the clone involved, myelomas may produce excessive IgG, IgA, IgM, IgD, and IgE. Next is as Waldenstrom. Macroglobulinemia is a condition where plasma cells can produce large amount of IgM, leading to increased blood viscosity. Thus, while immunoglobulins are critical for normal defense, their overproduction or abnormal production can also indicate diseases. Next is major histocompatibility complex, that is MHC. It plays a fundamental role in immune recognition. MHC molecules are highly polymorphic glycoproteins encoded by a cluster of genes and are responsible for presenting antigens to T cells. Without MHC, T cells cannot recognize foreign antigens, meaning adaptive immunity would not function. There are two types of MHC classes, MHC class 1 and MHC class 2. MHC class 1, it is composed of heavy chain, that is 43 kDa, non-covalently linked to a smaller protein, and B2 microglobulin. The heavy chain is folded into three domains, that is alpha 1, alpha 2, and alpha 3, with alpha 1 and 2 forming the peptide binding cleft. These molecules are expressed on almost all nucleated cells. They present peptides derived from endogenous antigens, that is protein synthesized within the cell such as viral protein or tumor antigens. Cytotoxic CD8 plus T cells recognize these peptides and kill the infected or abnormal cells. Importantly, MHC class 1 molecules are the principal antigen responsible for graft rejection since they differ widely between individuals. Next is MHC class 2. It is composed of two chains, that is alpha and beta, 34 kDa and 24 kDa respectively, each folded into two domains, alpha 1, alpha 2, and beta 1 and beta 2. It is found primarily on professional antigen presenting cell, that is APCs, dendritic cell, macrophages, and B cells. They present peptides derived from exogenous antigens, that is proteins taken up by phagocytosis or endocytosis. CD4 plus helper T cells recognize this peptide MHC2 complexes, leading to T cell activation and subsequent activation of B cell, macrophages, and other immune responses. Class 2 expression can be upregulated by cytokines, especially interferon gamma. Thus, MHC molecule act as the platform on which antigens are displayed, ensuring T cells can recognize whether the body is dealing with an infection inside a cell, that is class 1, or outside the cell, that is class 2 MHC molecules. Next is T cell receptors. T cell recognize antigens through T cell receptors, that is TCRs, which are always membrane bound and never secreted. Each T cell expresses a unique TCR generated by gene rearrangement, giving immense diversity to the T cell receptors. TCRs exist in two main forms, that is TCR2, that is alpha and beta, which is major type. It is composed of alpha and beta chains. It is found on the majority of T cells in circulation. It recognizes peptides antigen presented by MHC molecules, and it is responsible for most antigen-specific adaptive immune responses. Next is TCR1, that is gamma and delta T cells. It is composed of gamma and delta chains found in smaller numbers, mostly at mucosal and epithelial surfaces. These cells are considered more primitive and bridge innate and adaptive immunity. They play an important role in mucosal immunity and rapid defense against pathogens at barrier sites. TCR complex. Each TCR is closely associated with the CD3 complex, which transmit activation signals into the T cell when the receptor binds to a peptide MHC complex. This signaling event is what derives T cell activation, proliferation, and differentiation into effector or memory cells. Thus, TCRs are the antigen recognition machinery of T cells, with gamma and delta T cells contributing to rapid innate responses, while alpha and beta T cells mediate classical adaptive immunity. B cell receptors. B cell recognize antigens through their B cell receptors, that is BCRs, which are essentially membrane-bound immunoglobulins. Each BCR is made up of two identical heavy chains and two identical light chain, 2H plus 2L heavy and light chains, forming an antigen binding structure similar to antibodies. The variable regions of the chains determine the specificity for a particular region. Unlike TCRs, BCRs can recognize native unprocessed antigens. They do not require antigen presentation by MHC molecule. However, BCR structure are not sufficient by themselves, are not recognized Each BCR is made up of two identical heavy chains. One second, sir. So B cell recognize antigen through the B cell receptors, which are essentially membrane-bound immunoglobulins. BCRs are not sufficient by themselves to transmit activation signals. They are associated with Ig alpha and Ig beta, two transmembrane signaling molecules. When the BCR binds an antigen, Ig alpha and Ig beta transmit signals into the cell, leading to B cell activation. Once activated, B cell can differentiate into plasma cells, which secrete soluble antibodies, or into memory B cells, which ensure rapid responses upon re-exposure to the antigen. Thus, BCRs serve as the frontline detectors of antigens for the humoral immune system. Next is antigen specificity of Ig. Finally, immunoglobulins themselves are antigenic and exhibit different level of specificity. They can be divided into three types, that is isotypic specificities, allotypes, and idiotypes. So isotypes, these are antigenic determinants present on all immunoglobulins of a particular class with a species. For example, all humans have IgG, IgA, IgM, IgD, and IgE classes. Isotypic markers distinguish one class from another. Allotypes, these are genetic variations found within the same species. For example, not all humans carry the same allotypic markers, such as GM markers or IgG heavy chains. Allotypes are inherited and vary between individuals and populations. Clinically, they are important in transplantation and paternity testing. Idiotypes, these are unique antigens determinants located in the variable regions of immunoglobulins, particularly in the hypervariable CDR regions. Each antibody produced by a single B cell clone carrier is a distinct idiotype. Because idiotypes are unique, they themselves can act as antigens, stimulating the production of anti-idiotype antibodies. These create an additional layer of immune regulation, sometimes described as an immune network. So in short, immunoglobulins are not only effectors of immunity, but are themselves highly diverse molecules with structural variation at the class, genetic, and clonal levels. Summary for antigen. Antigen, any substance that provokes an immune response, recognize specificity by BCR on B cells or TCR MHC complex on T cells. B cell recognize soluble or free antigens. T cell recognize peptide MHC complexes presented on APCs. Second is epitopes, that is nothing but antigenic determinants. It is smallest part of antigen recognized by receptors. BCR, it recognizes soluble, surface bound or fragmented epitopes. TCR, it recognizes peptides plus MHC complexes. It is different lymphocyte can target different epitopes on the same antigen. Functional categories of antigens. Immunogens, it is complete antigens, it induces immune responses. Haptens, it is small molecules, antigenic but not immunogenic alone. Example is penicillin. Tolerogens, it is usually self-antigens, it induces tolerance and prevent response to self. Classification of antigen by origin and immune response. By origin, it is microbial. It is divided into different parts: febrile antigens, somatic antigens, flagellar antigen, and capsular antigens. By immune response, it is T-dependent antigens and independent antigens and dependent antigens. In independent antigen, it will stimulate B cells without T cell help. It large molecules with repeating epitopes. Example is LPS and polysaccharide. It produce mainly IgM weak memory. For T-dependent antigens, it require T cell help. Proteins, RBC antigen, haptens. It produce different types of immunoglobulins, long memory. Antigen determinants of immunogenicity. It depends on several factors: molecular weight, chemical complexity, foreignness, epitopes, host genetics, enzymatic susceptibility, its dosage, route and timing, and last is adjuvants. Antigen receptors, adaptive immunity and innate immunity. Antigenic specificity based on stereochemistry of antigenic determinants. A single chemical group or position can determine its specificity, and it is not absolute, so cross reaction may occur. Types of specificity. There are different types of specificity: species specificity, isospecificity, auto specificity, organ specificity, and heterogenic specificity. Summary for antigen recognition molecule. Immune system must distinguish self versus non-self. Molecules involved in antigen recognitions, that is antibodies, immunoglobulins, T cell receptors, MHC molecules. It is further divided into two classes, class 1 and class 2. Antibodies, humoral immunity, definition of antibodies, structure of immunoglobulins, and its proteolytic digestion through papain and pepsin, variable region, constant region, classes, different classes of immunoglobulins, that is IgG, IgA, IgM, IgD, IgE, abnormal immunoglobulins, classes of major histocompatibility complex, class 1, class 2, and T cell receptors and B cell receptors. So that's all for today's class. For questions, 3-mark questions. First, define an antigen. Second, differentiate between immunogen and heptane. Third, what are epitopes, that is antigenic determinants. Fourth, mention 2 examples of T-independent antigens. Fifth, state the main antibody class produced in primary immune response. Sixth, name the 5 classes of immunoglobulins. Seventh, what is the half-life of IgG and its special property during pregnancy. Eighth, define tolerogen with an example. 9, mention any 2 determinants of immunogenicity. 10, what are pattern recognition receptor, that is PRRS. give one example. For 5-mark questions, first differentiate between T-dependent and T-independent regions, antigens. Second, explain the factors that determine immunogenicity of an antigen. Third, describe the structure of an immunoglobulin molecule with its region, that is Fab, FC, variable and constant domains. Fourth, discuss the structural and functional differences between IgG and IgM. Fifth, write short notes on secretory IgA, that is SIgA, and its function. Six, explain the major histocompatibility complex, that is MHC molecules, class 1 versus class 2. 7, differentiate between isotypic, allotypic, and idiotypic specificities of immunoglobulins. 8, explain heptane carrier relationship with an example. 9, describe the role of PRRS and PAMPS in innate immunity. 10, write a note on abnormal immunoglobulins, Benzie-Jones protein, myeloma, Waldenstrom, macroglobulinemia. So with this, we completed antigen and antigen recognition molecules. Sir, are you there?
Yeah, madam. Lot of stress I think you have taken, madam, to cover immunology. Okay.
Yes, sir. No issues, sir.
Very nice presentation.
Yes,
I
sir.
think this presentation, no,
Yes, sir.
it will be beneficial for diploma, BSc,
Correct, sir.
and MS people also.
Messi as well. Yes,
Yes,
sir.
and that too I have seen, no, from
Yes, sir.
starting to now,
Yes, sir.
wherever parasitology is there, you have
Yes,
put together. That's what I
yes,
have said. So bacteriology means it is
yes,
put together as a bacteriology.
yes, sir.
So it is like package.
Yes, sir.
If they search parasitology, they get only parasitology details of explanation regarding parasitology.
Correct,
Very
sir.
nice, madam. Very nice, madam.
Thank
Very effective class. Okay.
Thank you,
Yeah.
sir. I actually
Thank you.
wanted to explain summary in more detail, but
Mm.
I have some work. That is why, in short, I explained. But
No, it's good actually. It's good.
summary, in short, I explained. But whole content, I tried to cover everything.
Yeah. I have seen. I have seen.
Thank you, sir.
Thank you. Thank you so much, madam. Thank you so much. Shall we wind up, madam?
Yes. Oh, yes, sir.
Okay.
Thank you, sir.